Welcome to IZON Science's webinar on characterization of extracellular vesicles from biological fluids. My name is Dave Yamani, and I'm the marketing consultant for IZON Science, and I'll be your host today. Extracellular vesicles have emerged as an important mediators of intracellular communication are involved in the transmission of biological signals between cells to regulate a broad range of biological processes. In light of the exponential growth in using extracellular vesicles for research and therapy, accurate methods to characterize their properties are needed. Today, you will hear two presentations about the characterization of extracellular vesicles from biological fluids. We're pleased to have Dr. John Ludlow from Zen Bio and Dr. Anup Paul from IZON Science join us today. Time permitting, we will conclude today's webinar with a brief question and answer session at the end. Our first presentation today, rapid isolation, purification, and detailed characterization of extracellular vesicles from biological fluids will be given by Anup Paul. Our second presentation will be given by Dr. John Ludlow and is titled Large-Scale Manufacturing of Microvesicles, In-Process Testing Using the Q-Nano. Before we begin, let me invite you to submit questions using the Q&A menu on the upper left side of your screen. To ask a question, select Q&A, type the presenter's name and question into the Q&A window, and press send. Any questions that we do not get to will be answered privately in a follow-up email message. Our first speaker today is Dr. Anup Paul. Anup is currently Chief Scientist, Americas Region at IZON Science. There, he contributes to IZON Science's research activities in the area of exosome and vesicles for diagnostic and therapeutic application, and works in close association with IZON Science's research partners. Prior to joining IZON Science, Anup finished his PhD in biomedical engineering and biotechnology from the University of Massachusetts. His work focused on evaluating health effects of nanoparticles and understanding nanobio interactions. Anup then went on as a postdoctoral research fellow at Harvard University, where he focused on studying environmental and health effects of exposure to engineered nanoparticles. His work has led to several publications in high-impact international journals. The title of Anup's presentation is Rapid Isolation, Purification, and Detailed Characterization of Extracellular Vesicles from Biological Fluids. Anup?
Thanks a lot for introduction, Dave. In today's webinar, I will cover some of the important concerns in the field of extracellular vesicles, utility of precise and accurate EV characterization, and IZON's integrated platform to tackle these issues. So, as is indicated by this slide, it shows there has been a considerable increase in publications related to exosomes, microvesicles, and extracellular vesicles over the years, and this reflects the trend. There is an intense research ongoing in the field of EV. Precisely, EV hold a lot of promise from diagnostic and therapeutic standpoint and have already led to some unique understanding of fundamental biological process. As we move on, we will see a lot of activity itself on pathological understanding of diseases which have been attributed to EVs. Now, the basic question is, what makes EVs so interesting and at the same time so challenging to study? Now, this figure, it highlights the variability in extracellular vesicles. Overall, there is no concise definition in terms of like the size and what would be the precise boundary of where all these three subsets will overlap. But in broad terms, there are three categories which EVs or extracellular vesicles are defined. Exosomes, they are on and around 50 to 100 nanometers in dimension in diameter, with the bilayer of phospholipid, and these are released by exocytosis of multivesicular bodies. Whereas microvesicles, or sometimes referred as microparticles, are a little bit bigger in size, in diameter, and are generated through budding of the plasma membrane. On the other hand, the larger ones, even in the range of a micron to 5 micron, are apoptotic cells, which are released by cells or tissue which is undergoing apoptosis. Now, from where it generates a lot of interest is that EVs have been shown to play a broad role and have diverse function in physiological as well as biological processes. So it makes them quite interesting from the standpoint of understanding the basic biology. Now, there is second thing which is important, that EVs have been linked to different functions when it comes to physiological condition, that is where everything is working well and fine, whereas they have different role or they have different information when there is a clinical disease or manifestation of a disease. So it makes them quite interesting from biological perspective, and they have several applications from diagnostics to therapeutic potentials. Now, in terms of what would be the challenges, as one can see, there is a huge variability in size as well as what is being released from the cell, and there is no definite or precise range that particles can be released in. So there is a heterogeneous population, which leads to a huge diversity in biophysical properties of these particles. So the key in turn would be to isolate and purify extracellular vesicles from a lot of biological fluids. Second thing is there are a lot of properties, which I would say biophysical properties, that define these EVs. There is a great need to link the size, charge, or the diversity of these EVs to the function they play, to the role they have in the biological, overall biological processes as well as the system. And then there is a grave need to do EV characterization, because that's how you will get to know what is the composition as well as what is the physical properties of EVs. So these two things are critical and current need in the EV field. Moving on, let's discuss some of the biophysical properties that are of utter importance when it comes to this particular field in exosome and vesicles. The cartoon over here from one of the papers, I have used this over here. It's a representative picture of an EV. And as you can see, the variability will come from almost everywhere. It can be within the what is in the core of EVs, which can be proteins, which can be DNA, RNA, or can be sorts of other biomolecules. On the other hand, on the surface can be different proteins, which are the surface proteins, surface markers, as one would say, as well as the variability in the phospholipid, which can be representative of where these EVs are coming from, and at times might have different charge as well. So in terms of applications which one is looking at is maybe biomarker analysis, diagnostic. Some have shown even the biosensing applications, and then there is this therapeutic potential, which is using EVs to deliver specific cargo. Now, all that would actually boil down to what are the properties of these EVs. Now, I have broadly classified them into three distinct properties. One is the size and size distribution of these particles. So as much as is the variability, you need to know the variability as well as the variability in the concentrations of each size as well, because that what defines what is being released, as well as in terms of delivery would be what is the dose which is delivered to, say, a specific cell or a tissue. So there is importance of understanding the size. That's critical. Now, the other thing would be surface properties, which in turn are dictated by the phospholipid layer, as well as what is in the surface, which is all the proteins which are embedded in the surface, as well as what is being linked to the protein on the protein itself, which might give you a different answer in terms of charge analysis, as well as speaks volume about what's going on the surface of these EVs pertaining to the role they play. There might be adherence of these particles, might be release of certain components, release of cytokines, and intracellular signals. So all sorts of functions are dependent on actually the surface of EVs. Now, something else which can be of importance is the morphology or the shape of these EVs, and the density, which might actually say again from where they are originating, as well as what is the content in them. So these are some of the critical biophysical properties one has to be aware of when it comes to extracellular vesicles. In order to answer some of the concerns which are ongoing, IZON has developed an integrated platform approach, which involves rapid EV isolation of extracellular vesicles, followed by a detailed characterization. As I have summarized in this particular slide, at the moment there are two particular route, I would say, the schemes which are being used for isolation as well as characterization of EVs. Broadly classifying, there would be biological fluids, especially urine, plasma, serum, or CSF, from which extracellular vesicles are being purified or being isolated. At IZON, we have developed a QEV column, which is a size exclusion chromatography column, and that leads to purification of EVs within 15 minutes, followed by detailed characterization using QNano, TRPS analysis. TRPS stands for tunable resistive pulse sensing. So that's scheme one. Second scheme is most of the time cell culture are being used as a much more refined option in terms of control, what is being released, what is being studied. So instead of the complexity possessed by a biological system, it is much more simplified in that way. So most of the time, since cell culture volume, we are dealing with a larger volume, what we can do is we can have a pre-step in which we do a high-speed centrifugation, getting rid of all the larger cell debris and particles which are actually not part of the EV, I would say, size range. And then using the pellet, a step two, using the pellet for QEV protocol, the output, the fractions which are collected are purified, and then as in scheme one, can be further evaluated in terms of size, concentration, and charge through using a QNano. I have referenced all the papers which are key papers in terms of methodology which has been applied or adopted by the users using QEV as well as QNano for purifying as well as characterizing exosomes. And interested people can actually go and look at the details in there, as well as go on the website, IZON's website, under publication section, and get more information there. To briefly summarize some of the key benefits of QEV, which is a size exclusion chromatography, is that it's a fast, rapid 15-minute isolation protocol, and it gives a purified sample. That is, it removes almost 99% of contaminating background proteins, can be lot of biological biomolecules if you are using biological fluid, say serum or plasma. It removes up to 95% of contaminating HDL, that's high density lipoproteins. And one of the key and the most important thing which it does, it maintains the integrity of EVs and the function of these extracellular vesicles if somebody is using them for further analysis and especially for therapeutic applications. So that would be a key. Now, some of the important benefits which have been time and again shown by several publications related to QNano is that it's an accurate and precise particle by particle exosome and vesicle characterization technique, and gives you a precise size and size distribution. Given it's a direct counting, it gives you a reliable EV concentration and an accurate analysis of total population of EVs in there. And third, which has been shown quite recently by extensive publications which have been on the charge through QNano, is the detailed particle by particle charge analysis of nanoparticles as well as EVs. So to exemplify some of the potential of this integrated platform, what I would do is I would highlight some of the ongoing projects, collaborative projects with the academic partners which IZON has, and highlight some of the ongoing studies as well as the results which we have achieved, which shows the utility of using QEV in conjunction with QNano. So let's look at the project with Professor Nigel McMahon's group at UNC Chapel Hill. This project is related to tissue factor particle. In a brief overview, I would say tissue factor is a transmembrane receptor, transmembrane receptor that binds to factor 7A, and triggers blood clotting after a vascular injury. These two factors combined gives the procoagulant activity to tissue factor particles. Now, tissue factor particles are released in higher number or amount and have been shown for certain cancers, especially when it comes to pancreatic and brain cancer, that the release is quite high. So there is a definite interest in looking at these particles and what are the biophysical properties of tissue factor particles. To point out, CD24, CD43 are the important adhesion surface proteins which are in the membrane of tissue factor particles, and MYC1 is the tumor marker. For this study, as I mentioned, this is ongoing. This work has been done in past few weeks, and what we have done is we have used the integrated platform for looking at tissue factor particles released from plasma, which is LPS induced, as well as cell culture samples. In here, two pancreatic cell lines were used, one which is known to have a tissue factor positive particle release, which is BXPC3 cell line, as well as one which is tissue factor negative, which has a minor tissue factor activity, that's PANC1. Now, as a functional endpoint to what we are doing, we use tissue factor activity assay, currently being used in Professor Nigel McMahon's group, as a functional endpoint to the measurement which are done through QNano. There is a certain utility in normalizing tissue factor activity as an output in conjunction with TRPS. And a final point which I would like to exemplify is the approach which we are using to capture specific tissue factor particles as a subset population from the whole population of microparticles, followed by TRPS analysis and looking at some of the interesting results which have been generated in the meantime. Now, to point out some of the basic findings which came from this study, this is figure 1 on TRPS analysis on PANC and BXPC3 microparticles. This is without QEV step, but what it shows is there is a substantial population. First of all, there is a variability in both the cell lines when it comes to size as well as the number of EVs which are microparticles which are being released. There is a substantial population which actually lies in the size range of 100 to 300 nanometers, and there is a minor population which is a trailing population, as I would say, from 500 to 1000 nanometers. So indeed there is a diversity in what is being released from these cell lines, as well as there is variability. PANC1, which is TFMP negative cell line, has almost 3 to 4 folds higher concentration of microparticles in comparison to BXPC3 cell line. Now we used QEV to purify the samples which are from plasma as well as from cell culture. On the x-axis, you can see this is the output, which is what I'm comparing is microparticles which are released from cell culture as well as microparticles which have been post purification using QEV from plasma samples. On the x-axis is the size, which has been evaluated by QNano, and on the y-axis is the concentration of each of the particles as being released. Now, there are certain utility when it comes to analyzing size and concentration, as I would exemplify that when you are comparing cross comparison of samples, it is of high utility that the comparisons are made not just on the basis of what is the size range as well as what is the output concentration. They have to be broken down into subset as well, that what is the size range in particular bin. Say in this case, if I'm looking at a size range from 100 to 1000, there is, of course, there is variability when it comes to in the number, that the output concentration is different. But when we break down even precisely the concentration fraction from say 100 to 300, there is a definite information here, as well as from C300 to 1000, that there is a very minor population from plasma which is being released upwards of 300, whereas there is a lot of microparticles released from cell culture. So if we are cross comparing samples, it is of importance to have a precise as well as high resolution characterization of particles, as well as the utility when it comes to looking at specific subset of population as well as the concentration fractions. QNano gives you all this freedom to actually compare samples not only with size as well as size coupled with concentration fraction analysis. So definitely we had a smaller population as indicated by this particular figure. Moving on, as I said, maintaining the function or what is being collected post QEV purification, there is a critical need to see that the particles have maintained the integrity as well as maintain their functional activity. In this case, what we did was we collected different fractions and did tissue factor activity assay, which speaks about the functionality of these particles. As is indicated, column 1 represents different QEV fractions. Each fraction is 0.5 ml in volume. Column 2 represents BXPC3 particles. Column 3 is PANC1. So as it indicated over here, fraction 4 to 6 and 7 to 9 are the ones which had saturated signals. So they had a huge tissue factor activity. whereas the other ones had really minor components. So in essence, we were able to purify samples, especially in these fractions, which gives you higher activity as well as they are concentrated in number. In comparison, PANC has a very minor activity, but again, it was in these two fractions. And as we have seen previously, most of the sizing is between 100 to 1000 particles, nanometers, which is being seen repeatedly for these particles. One thing which I would like to point is using IZON TF spiked samples, negative samples especially, there was no enhanced TF activity, which is tissue factor activity, was observed, indicating the fact that we were able to actually purify the particles from the IZON TF spiked or the floating IZON TF molecules there. Moving on, this is a basic, or I would say this is an overview slide, which is a simple approach for exploring the utility of capturing specific tissue factor particles on magnetic beads, which are conjugated with specific antibodies, and analyzing them through Q-nano. So what we have done is we have characterized the input, in case what is the size as well as the concentration of magnetic beads bound antibody, which we are that's input A, and then tissue factor microparticles, that's input B. We mix them, we can actually run Q-nano on them to see what's going on there and how much is actually the total concentration, followed by a step which is specific binding. This is for 2 to 3 hours mixing, and followed by a mag separation step in which particles which are bound to magnetic bead are selectively captured, whereas the ones which are unbound are in the supernatant. We do a washing step and again collect the supernatant. In essence, supernatant represents what is unbound, whereas the one bound to magnetic bead would be the pellet. Let's look at some of the results. Here, this is tissue factor activity assay as done on the input, which is the BXPC3 particles, and as done on the supernatant, as pellet had there's magnetic bead, so it didn't it had its own artifacts. So we just focused on looking at input, which is BXP3, and the supernatant, which is unbound BXP3. As is indicative here, these numbers are generated using Q-nano, and what we have done is we have used number as an input, and this can be done for any other assay where particles are being used, and there is a determined activity in picograms per ml, and what is being left, that is what is measured in supernatant. As it indicative over here, as we increase the number of BXPC3 particles, which is TFMP positive particle, keeping the magnetic bead particles constant, we see there is increase in supernatant TF activity, but as compared to what was being input, we see almost 60 to 70% of particles being captured as we increase the BXPC3 concentration. As a control, what we did was we increased even the magnetic bead concentration 10-folds to see if we can capture more, and it turns out that we were able to, as indicated by these results over here, we were able to almost capture 90% of the population through this approach. So this is quite promising, and to match it with TRPS analysis, what was done, we looked at the pellet, as binding will lead to increase in volume as well as increase in number of particles, which are actually resulting in the pellet itself, supported by the fact that what we see in tissue factor activity assay. Similar, we looked at the size as well as concentration of pellet using TRPS. X-axis represents the size and Y-axis represents the concentration. As one can see, there is a proportional increase in number as well as at times increase in the size of particles after binding. So this is pellet, so this is BXPC3, which is bound to magnetic bead. This red bar over here indicates the master mix, which was for higher magnetic bead. So you can see it's quite precise and as well as of high resolution to do all these sorts of analysis on Q-nano. Moving on, the TRPS output can be actually converted to volume, and one of the function is looking at percent volume as being output from the software itself. Looking at the differences in volume for pellet as well as supernatant, it's indicative of what was seen in tissue factor activity assay, that as we increase the number of BXPC3 particles, there is substantial increase in the pellet volume in comparison to just the control magnetic bead. In accordance with, there is a decrease in supernatant volume as well, which is in comparison to BXPC3. So this is of utility because it can be used as a parallel approach, not to just look at size, concentration, but even to look at volume, because that might be more indicative of the overall complex in the behavior of these particles when they are interacting to each other. So what essentially one can do is break down a biological process into much more of a mathematical model, or use numbers to see as one sees in chemistry. So that's a neat approach to look at particle behavior. So in second study, this is with Professor Dimitrovello at UMass Lowell. They're working on urine samples which are collected post occupational exposure. It's a pretty neat study in which exposure is measured. Ambient particles are measured in air, as well as the samples, human samples, in this case urine, is taken from the workers, and then IZON's integrated platform is used for purification as well as analysis of EVs. In this case, the real potential is the development of a non-invasive method for potentially understanding biokinetics of exposure to isocyanate, because isocyanate is that's the toxicant of interest over here. So just a slide on this particular study, and it again highlights the utility of looking at samples and cross comparing them, say, exposed to control. Now 2 points which I would like to emphasize over here is, in comparison to control, one can see that exposed have they have different size profile. That is, there is more of smaller population as being seen in EVs which are released in urine of these exposed people in comparison to control. Second, there is almost 10-fold increase at times in the population of EVs, which is indicative of what is being released in the urine post exposure. These are critical indicator, and it's a first impression of what's going on, and if there is a real need to even study further. And moving on, building on the initial results on this part, there are biological assays which are being looked at, looking at cytokine as well as microRNA, and how they link back to known biomarkers of exposure, in this particular case isocyanate. So to exemplify, yes, this QNano gives you the power to get a holistic approach on what's the physical properties of particles, EVs, in different biological systems. So in this particular slide, this would be interest to people who are interested in quite keen on the surface properties of EVs. So this is identification of specific surface proteins on EV surface. As highlighted in the figure, binding of a ligand, which is in this case is a CD63 aptamer, which will potentially bind to CD63 proteins or surface marker on surface of these EVs. So post binding, there can be increase in size, as is indicative over here. This is green indicates plasma EVs, whereas red indicates EVs plus CD63 aptamer. So a binding event can lead to increase in size, as well as decrease in this case is decrease in charge. But as one can see, there can be two output. One can be size and other can be charge. So in cases it can be either of size or charge, or it can be both. And this gives you a broad view of looking at the surface of these exosomes. And in essence, TRPS can be used for analysis of specific EV surface proteins. So we are building on this work and looking at other different subtypes of population, and is of utility when you are looking at different surface markers and trying to see what is the global population as well as what is the subset of population represented in each fraction. So clearly, TRPS is of utility if you have size and coupled with charge and concentration. To highlight the final study, which was published recently from UK, Mark Plett's group, they have used TRPS as a biosensing tool and that's quite remarkable to do because they have gone down to nanomolar concentrations. The cartoon over here highlights the overall approach. In this, magnetic beads are coated with a thrombin aptamer, and another set of polymeric beads are coated with its complementary DNA strand. When these two particles are mixed or when they are bound, it leads to a micron size aggregate, or this micro size particle, which would in turn be, as indicated by TRPS input, this is a bigger blockade signal. Now, on addition of thrombin, which is the antigen over here, it breaks, it binds to binds to the specific antithrombin aptamer and leads to release of or dispersion of these complexes. Since we have magnetic beads here, so magnetic beads can be specifically captured out, and both population can be measured, which is resulting polymeric bead as well as magnetic bead itself, and gives you a precise understanding of what's going in the overall system. And this can be used to design successful assays, as well as highlights the utility of TRPS as a tool for biosensing applications. To conclude, there are two major, based on the data which I have shown, there are two major conclusions. One is QEV coupled with QNano as an integrated platform provides a rapid EV isolation and comprehensive high resolution EV characterization, and this done in conjunction can be performed within 30 minutes to go from EVs in plasma to EVs purified and to EVs well characterized. So that's a whole lot of information within half an hour in comparison to other competing technologies out there. And this integrated platform has tremendous potential for biosensing and EV surface marker analysis. I would like to acknowledge the tissue factor work done with Professor Nigel Mackman's group and Yohei. They have been quite helpful and have provided insightful comments and feedback. I would like to acknowledge Professor Dimitar Bello and Ekjot Singh, his student, for collaborative work on EVs in UREM, as well as IZON Science team. Thanks a lot for.
Thanks, Anoop. Our second presenter today is Dr. John Ludlow. John is currently Executive Director of Regenerative Medicine at Zen Bio Incorporated. Prior to joining the company in 2012, he was Senior Director of Process Research and Assay Development at Terragen, a tissue engineering and regenerative medicine company located in Winston-Salem, North Carolina. John has also served as Senior Director, Cell Therapy Program at Vesta Therapeutics Incorporated in Research Triangle Park, North Carolina for 5 years. John has developed and managed research and preclinical programs, initiated clinical trial sites, and directed development activities for cell therapy and tissue engineered products. John started his career as a faculty member at the University of Rochester Cancer Center, New York, where he maintained an independently funded research laboratory training graduate students and postdoctoral fellows in the area of tumor suppression gene expression, protein structure, and function. The title of John's presentation today is Large-Scale Manufacturing Microvesicles in-Process Testing Using the Q-Nano. So John, is it down in your lower corner there?
Yeah, hold on. Yeah, okay, yeah. Technical difficulties, all right. I'm not seeing anything.
Yeah, so you probably want to go to share your desktop, and then you want to go to the you can then go to the PowerPoint presentation so you can put in presentation mode. There you are. Okay.
Okay.
So now go share. There you go. Okay, excellent.
Sorry about that, everybody. Okay. Hey, thanks a lot, Dave, for that introduction. As Dave mentioned, my name is John Ludlow, and I'm the Executive Director of Regenerative Medicine at ZenBio. And I want to welcome everyone to this webinar titled Large-Scale Manufacturing of Microvesicles in Process Testing Using the Q-Nano. ZenBio has been in the business for over 20 years with a continued focus on helping our clients build greater capacity for preclinical screening, drug discovery research, tissue procurement, cell isolation, consulting, and custom assay development. As part of our ongoing efforts to stay current with the needs of our clients, our interest in microvesicles is from a therapeutic viewpoint, more precisely the production of these nanoparticles in a manner consistent with regulatory agency guidelines. Part of these guidelines include something called in-process testing, whereby at specific steps during the production process, samples need to be taken and analyzed. We're using IZON's Q-Nano to analyze microvesicles during our production process. Now, in the previous talk, Anoop gave a very, I think, comprehensive overview of the capabilities of this instrumentation. I'm going to step it back a little bit. During my talk, we use the Q-Nano for in-process testing for really two parameters. First, what size are our particles? And second, how many of them do we have? So to provide some rationale or background into what I'm going to present, I'm going to start with the key raw material for our microvesicle production process, multipotent stromal stem cells that can differentiate into a variety of cell types, the so-called mesenchymal stem cells. This differentiation ability makes these cells an obvious choice for development of tissue engineering and regenerative medicine therapeutics. Regarding their mode of action, it has been demonstrated that MSCs do not repair tissue and organ defects by necessarily differentiating into the desired tissue type, but they act more in a regulatory role. This paradigm shift has come about following demonstration that MSCs can inhibit apoptosis, they can stimulate angiogenesis, promote endogenous cell proliferation, and also inhibit inflammation during tissue regeneration. Accomplishment of these functions is through growth factor and cytokine secretion, the paracrine effects, in addition to cell-cell interactions. This paracrine hypothesis introduces a fundamentally different dimension to the therapeutic applications of MSCs in regenerative medicine. By replacing transplantation of MSCs with administration of their secreted microvesicles, of which exosomes are the subclass most researchers are familiar with, many of the safety concerns and limitations associated with the transplantation of viable replicating cells could be alleviated. While exosomes as a therapeutic have been tested as a form of cancer vaccine in the clinic, these tests were limited to particles produced during short-term ex vivo culture of autologous dendritic cells. The take-home message from this study, however, is that exosomes from these cells were deemed safe in the small clinical trials that were conducted. Unfortunately, the process by which these exosomes were manufactured can't provide guidance for regulatory compliance, large-scale production of exosomes, which can be used in more comprehensive clinical trials for other indications. The needs for such a product include the following: a more physiologically relevant cell culture environment for microvesicle exosome productions. Stated another way, three-dimensional culturing instead of two-dimensional. I'm sure we're all familiar with the arguments regarding 3D culturing as being more in vivo-like, as this is how tissues are set up. The idea here is that microvesicles harvested from these 3D cultured cells will be better than those from cells cultured in 2D. To my knowledge, however, this idea hasn't really been formally tested. A scalable platform for efficient and robust microvesicle exosome production. We'll show some data comparing yields from both 2D and 3D methods of producing microvesicles from cultured cells. A CGMP-compliant closed cell culture system for manufacturing. This can be a limiting factor for large-scale production of just about any biologic for therapeutic use. Now, if there is no way that the product can be produced in a closed system, this needs to be convincingly explained to the regulatory agencies. If there is at least some way to do so, however, then you may be obliged to try and figure this out and use the technology. We at ZenBio believe that the needs summarized on this slide can best be addressed using hollow fiber bioreactors to culture cells from which conditioned medium containing microvesicles can be harvested, isolated, and concentrated. This is a schematic of how the system works. This picture is taken right from the FiberCell website. Starting at the top right side of the diagram, medium flows from the reservoir bottle by means of a pulsatile perfusion pump. The bottom right, through the oxygenation circuit, bottom left, where it's oxygenated and saturated with CO2 before it enters the bioreactor cartridge, which is at the top on the left-hand side. Cells growing in this environment are bathed in fresh medium as it is perfused through the hollow fibers, exiting the cartridge and returning to the reservoir bottle for recirculation. Metabolic waste is dialyzed away from the cells and into the circulating medium. There are sub-nanopore filters at either end of the cartridge to prevent cells, and as I will show you in a minute, microvesicles, from leaving the bioreactor and becoming diluted in the reservoir medium. Since we cannot directly observe the cells in the bioreactor, glucose consumption rate is monitored, as consumption of glucose is directly related to cell number, growth rate, and metabolic state. I personally use the AccuCheck blood glucose monitoring system. This was originally marketed to those with diabetes to monitor their blood glucose, and it's available at any pharmacy over the counter. So, restating back to the previous needs slide, we believe that FiberCell systems offer one of the better solutions. The bioreactor is a high-density, continuous perfusion culture system that more closely approximates the environment in which cells grow in vivo. The advantages include an extremely high surface area to volume ratio, 10 to 200 centimeters square per mil volume, thus making densities of 1 to 2 times 10 to the 8th cells per mil or more achievable, a density which is close to in vivo tissue-like densities. In addition, since the cells are cultured on a porous support, a condition much more similar to in vivo than two-dimensional culturing on a plastic surface, there's no requirement to passage the cells. The perfusion system maintains cell viability and relevant metabolism for extended periods of time, on the order of months. Once the cells are seeded into the cartridge, the system complies with the intent of the regulatory guidance for GMP manufacturing in that the system is closed, whereby the cells are not exposed to the atmosphere during production. So how do we isolate? Our microvesicle isolation procedure is on this slide. We use ultracentrifugation. There's several variations to this general scheme, and I'm not going to say too much more, other than several of our clients prefer this method over others. The sucrose cushion option is designed to further isolate smaller exosomes from the larger particles. However, in our hands, this additional ultracentrifugation step dramatically reduces our yield to unacceptable levels. So, here's where our in-processing testing begins using the Q-Nano system. And these pictures I show on the slides are just grabs from the final report that the machine presents you after the analysis. As shown here, fetal bovine serum containing medium is loaded with microvesicles in the 150 to 200 nanometer range, which we do not want to contaminate the microvesicles we want to isolate from the cultured cells. Now, there are some ways to discriminate between human and bovine microvesicles, for example, specific antibodies, magnetic activated cell sorting, but separating them is never complete, and having bovine contaminants in a potential clinical trial product should be avoided if at all possible. Again, just taken from the report, this slide summarizes the graphical data, and as you can see here, 5 times 10 to the 6th, 5.6 times 10 to the 10th particles per mil is what we found for the FBS-containing culture media. Now, as mentioned before, FiberCell system has filters at both ends of the hollow fiber cartridge, allowing smaller growth factors free passage across the filters, thus providing needed nutrients to maintain cell health, while preventing microvesicles from moving from the circulating reservoir medium into the bioreactor. Microvesicle exclusion is demonstrated here by filling the bioreactor with serum-free medium, circulating the serum-containing reservoir medium for 7 days, then removing the serum-free reservoir medium and assaying for the presence of microvesicles. And no, this is not an error. There really are no data points on this graph because the Q-Nano did not detect any microvesicles in the bioreactor sample. And with no data points, there aren't going to be any statistics. So, confident that our microvesicles of interest will not be contaminated with microvesicles from the FBS contained in the medium reservoir, which the cells need to maintain their health and vigor, the next step was to seed the bioreactor with 3 × 10⁸ cells. Once they became established, we replaced the medium within the bioreactor with serum-free medium and harvested after 48 hours to examine the microvesicle yield. The purpose here was to optimize production time. And again, shown in the statistics slide here, we isolated or recovered about 4.8 times 10⁹ particles. We repeated this procedure and harvested after 4 days, again to see if we could increase our yields. Distribution being shown here about 150 to about 250. And sure enough, we increased our yields, a 2-log increase compared to the 48-hour run. We decided to keep this harvest schedule for all subsequent production runs, as this also coincided with depletion of glucose in the reservoir medium, indicating that it was time to discard this bottle and replace with a fresh one. Coupling these 2 events seemed to be most efficient. This table summarizes the yields and totals from a 7-week production run using 2 bioreactors. I'll point out that the yields vary from harvest to harvest, as does the total protein yield, and we do not observe a correlation between protein and microvesicle number. I don't have an explanation for this. This is something we will look at in the near future as part of our optimization work. To help put this scale up in perspective, we compared media usage alone to a production run of 130 T225 flasks over a 7-week period. It needs to be appreciated here that continued passage was required to get up to 130 flasks, with medium changes every 3 to 4 days, and the flask could only be incubated with serum-free medium for 48 hours before they started to feel the effects of no serum in the medium. This limited us in our ability to collect microvesicles, as the cultures had to recover for 2 days in serum-containing medium before replacing with serum-free medium for microvesicle collection. With the bioreactor, we did not have to pass cells, nor did we have to starve them of FBS during microvesicle production. What I didn't include here is the amount of PBS needed to wash the 130 flasks, which was considerable, to remove all FBS and bovine microvesicles just prior to adding the serum-free medium from which we would isolate our microvesicles of interest. Finally, from the 130 T225 flasks, we obtained 16 × 10⁻⁻ total microvesicles, 2 logs lower than what we obtained using 2 bioreactors over the same amount of time and using less medium and hands-on manipulation. As a for-profit company, always keeping an eye on the bottom line, using less materials and less attention by personnel translates into a reduction in the cost of goods. In closing, this slide summarizes our efforts to scale up microvesicle production in a regulatory compliant manner for therapeutic and regenerative medicine applications, using IZON's Q-Nano as an integral part of our in-process testing to monitor the size and yields of the microvesicles during a production run. With that, I thank you for your attention.
Thank you, John. We'll now have a brief question and answer session. Subhash Kalari is helping me today and will be the Q&A moderator. Subhash?
Yes. Thank you, Dave. So the first question we have is from Lawrence. And the question she asked is, what are the key advantages of using QNano over nanosite instrument? Anup, do you mind answering that question, please?
Sure. Thanks for the question. As has been shown in actually a lot of publications over the years, that there are basic differences when it comes to measuring particles, and the most accurate is when it is done through impedance. Light scattering is what is being used with, I would say, nanosite and other light-based instrument. What the output is hydrodynamic diameter, whereas what is the output in QNano is a physical diameter, which is much more reliable and accurate, and it can go as below as just differences in few nanometers. So that is something which is of tremendous utility. Now, I would say the key things which one has to be aware is to have accurate, high resolution size, because size when coupled with concentration is a very meaningful index when you are cross comparing different samples, which is a basic output from QNano. Now, one of the limitations I would say when it comes to light scattering instrument is that there's limitation both at the smaller end as well as the larger end, because if you look at what is being calculated is the output intensity. So the larger particles will give you a larger intensity, which might lead to actually over calculation of or under calculation of the smaller particles. Whereas when you look at the smaller particles, on the other hand, it might have much more information being in comparison to what it should be overall the global distribution, gives you inaccurate answer. So at both ends there are problems. So that's there. And key thing which people have pointed out time and again is the settings. So you change the setting, you get a different answer, which is actually highly unreliable. So in essence, what I would say is that QNano gives you more accurate, and it gives you a direct counting, so that's even accurate in terms of number. And there are modules which can be actually used for multi-pressure analysis to give you even precise number, concentration, as well as perform charge analysis in conjunction with size. So that's a tremendous benefit over other characterizing equipments.
Oh, thank you for the answer, Anup. And the next question, what we have here is from Raul.
Mm-hmm.
His question is, how do you know that we have removed particles that are not bound to the exosomes? It seems that we remove a lot of stuff using simple technique SEC, but how do you know that we have not removed any particles that are bound to exosomes?
Right. That's a pretty good question. Now, I would say is that we have to look at actually what is out there and what are the other techniques which are people using, and when you compare to what is being used, the ones which are being used is either ultracentrifugation or filtration. The yield is quite low first, and second is the purity is not what one would like, especially if you look at the precipitation methods out there. So QEV is the single method which actually does a pretty good, I would say, the best job actually in terms when it comes to purification. Now, the question which I would say I think of is what is bound to these particles, which might be of interest themselves. Now, in terms of purification or say isolation, as has been shown in the articles recently by in Journal of Extracellular Vesicles by few groups, they've already pointed out that when you look at comparing in terms of like what is the surface marker of EV evaluation, you look at that, you can you use a secondary technique, which is basically a biological technique, as I have done in terms of tissue factor as well, to quantify what is the yield. And mostly it's pretty high. Say for the particles I was doing, we were getting close to 5e to the power 8 particles per ml of QEV fractions, which we used back then. Now, to answer what it removes, it would be proteins, as been shown in these publications, as I pointed out in my slide as well, that proteins being smaller in size are actually eluted later in the fractions. So the fractions which we use for vesicles and have been quantified, well characterized, as well as in terms of size profile, has been shown that fractions 7 to 9 are the one which have enriched population of exosomes, whereas the later fractions on and around 20 are the ones which have proteins. So in essence, you have actually remarkably removed all these biological proteins from vesicles. And in terms of, I would say enrichment, as has been shown, it is almost 6,000-fold enrichment. If you compare 7 to 9 fraction 7 to 9, you pull everything together, that fraction is enriched 6,000 times as compared to protein fractions if you look at the later point. So that's quite a bit of enrichment. And in terms of purity, you can look at protein gels as well as other endpoints which have been routinely done, and it does a tremendous job in purifying. Now isolation is a different thing. Now when it comes to purification, you might have to, from the whole subset or like the whole set of EVs, you might be interested in just the 5%. Now that's a different equation, and for that you need to have specific antibodies or aptamer approach, as I have shown, to quantify actually that if it is 5%, 10%, what is this of all the EVs, what is the EVs of interest one is looking at.
Okay, Anup, we have the next question is for John. John, we have a question for you, and the question is pretty straightforward. So the question is, have you tried using any of the exosome precipitation solutions for isolation from the bioreactors?
From the bioreactors, no. I have tried these solutions using tissue culture supernates from smaller tissue culture flasks like a T25 or a T75. Again, most of our clients seem to prefer the ultracentrifugation method. Apparently there's some concerns about contaminating the exosomes with PEG, especially if the exosomes are destined for preclinical or in vivo testing. Also, volume-wise, it's more cost-effective to go through the ultracentrifugation route. However, if your volume is limited and you do not have access to an ultracentrifuge, then the precipitation method is very useful. But short answer from the bioreactors, no, and it's primarily due to a volume issue.
Oh, thanks, John. And
yep.
So we have time for 2 more questions. And so let me look up. And just by the way, if you have any questions, feel free to put them on the Q&A box, and we will answer them by email if we don't have enough time. And next question is from Levine. Is there a way to separate the exosomes from other EVs with a similar size? Hello?
Oh, that's okay. Yeah. Well, depends. Like that's what I think the whole interest is. If you can you can separate populations of even different size as well as of the same size. Now it in turn would actually speak of what is in them. That is there a difference in density? Maybe there's a difference in density. Then you can have same size with different densities. Maybe there's difference in charge. So that has to be looked to, and that's where there is Q-nano, which is of utility, that you can look at the surface properties. So that can be an output as well to look at. So in essence, yes, there are ways to actually characterize. Now, in terms of isolation or purification of the subset, that's where I think it is much more tricky, because one has to look at what is the surface marker for that particular exosome, and that has to be derived before you actually can pull it out. So I would say that's where the field is building, that once you know what is the surface marker of interest, there are generic markers which one has looked at, and CD63, CD9, and they have pulled them out, and then looked at even much more representative markers for like the endpoint they are looking at. And once that is known or determined, say you have a biomarker of interest, then you can actually use that to pull out specific populations. So it's I think it's work in progress, but yeah, there are ways, and to look at subpopulation to characterize them. And I think biophysical properties are key when it comes to looking at the variability overall, like when it comes to, say, comparing control versus exposed. So yeah.
Okay, and one final question to John. It's from Tanya. So the question is, in terms of regulatory compliance with the FDA, what is your experience on source cells?
Sure. For with the projects that I've been involved in that have made it through and made it through IND submission, acceptance, and clinical trial, the MSCs that were derived from adipose tissue seem to have the least amount of questions raised when going through the FDA. Now, clearly, we needed to demonstrate the transformation capabilities of these cells. We had to characterize them with respect to specific markers, and we also had to do studies where we would, only use we only take the passages out a certain distance before we were no longer certain that the cells were no longer producing the materials that we needed them to. But again, I've had the least amount of questions raised when using MSCs derived from adipose tissue.
Oh, thanks for the answer, John. And I pass on to Dave, our host.
Thanks, Subhash. Well, that's all the time we have for questions. Unfortunately, we don't have time today to answer all the questions that were submitted. If you're still submitting a question, we will keep the webinar website open for 5 more minutes to allow you time to send it. Unanswered questions will be answered privately in a follow-up email message by John or Anoop. I'd like to invite you to visit www.zenbio.com for more information about ZenBio Incorporated. ZenBio is a leading global provider of advanced cell-based solutions and services to the life science, cosmetic, and personal care communities. Founded in 1995, their mission is to provide the highest quality human cell systems, reagents, blood products, and contract services while continuing to develop and commercialize innovative tools that accelerate discovery. I'd also like to invite you to visit www.izon.com to see IZON's list of publications and resources on the isolation and characterization of extracellular vesicles. To see them, select Publications from the upper navigation menu, then select the application area that interests you from the navigation menu below the picture. This concludes today's webinar. On behalf of John, Anoop, Subhash, myself, ZenBio, and IZON Science, I thank you for attending today's webinar, and we look forward to seeing you at future events. Have a pleasant day.